Today first part we will just recap a little bit of continuation from the last previous session, and then we will move to fiber photometry, which is taught by Atharva. So let's get started. Hope my screen is visible. Cool. Okay. So today we'll have a bit of recap from last lecture, and we'll see how a typical EEG spectrum looks like, and we'll deal with a special case of noise called as factoral noise or pink noise. So this is a topic for this first half an hour. Uh, so here from last class we have seen this ERP from each electrode, how you can visualize it. So from each electrode you plot one ERP averaged across trials of interest, and you get a ERP plot like this as shown here. Uh, so yeah, you can see my cursor or my writing, right? Yeah. Is it visible? Okay.
Yes.
Yeah. So this time course you see is a ERP from each electrode, and you plot it for all the electrodes. This is one form of visualizing ERPs. The other form is, okay, one second. Yeah. Other form is visualizing the ERP components using topography. So what do you do is in that ERP time course you have a selected window like this. Choose a small window around some centered point, and you can plot for each electrode what is that amplitude on a 2D map as shown here. So this is called topographic visualization of you can visualize anything, not only ERPs. You can visualize alpha power, spectral power, or any quantity of interest if you have across electrode wise metric. So this is another form of visualization, and one thing that we didn't deal in the last class is called ERP image. So instead of plotting average across trials, what you can do is you can plot a time course of every trial for a given electrode, for example, and you can stack it like an image. So yeah, so here you see, right? So here what you see on y-axis is each trial, and you plot the ERP, like you get one time course for each trial from an electrode. This electrode we are using, that is POZ, as you see here, and you plot across all trials. So each row on this image corresponds to one trial, and there is some kind of sorting happening. So we sort according to some events in the task or reaction times of the subject. So this black line you see, right? This black line here, this black line is nothing but sorted trials. So instead of plotting each trial sequentially, you sort it based on some metric on the task, and then you plot for each trial, and which gives an ERP. So so generally trial wise ERPs are very variable, so you don't, unless you average, you don't see it. But what is happening here is we take a smoothing window, and like we take a window like this and smoothen it and stride, like step it, and that way you get a nice ERP image like this. So this is another way of visualizing ERPs. This is called ERP image. Is everyone clear with how an ERP image is formed? Okay, cool.
Yeah, just
So
one question, Deepak.
yeah, sure.
What do we infer from the ERP image?
Okay, so here what in this case, in this visualization, we are losing on each trial what is happening. Like it's an average metric across a set of given trials of interest. But we also want how it is happening, or what events are happening across trial, and if you say, if you have some trial wise metric, how it is changing. So this can show trial wise dynamics here if you don't average. Rather, if you smoothen with small number of trials in this y-axis, you get an ERP image. So to get this sort of an image, you need some kind of sorting. You can't just plot a just set of ERPs on y-axis. So based on some metric, you arrange the trials, and then on each trial you plot and then take a smoothing window like this. So I'll tell you how this is useful in the next slide. So right now, if you're clear with visualization, I'll move forward. If any doubts like how we are constructing this, we can. Okay, sure. Yeah. So now here is one example of how ERPs can be used as signature of cognitive process. So this is the paper that from this paper I'm going to discuss some of the results. So here you don't have to make sense of this title. Just remember that it is saying something about perceptual decisions or decision making in humans. So here the task is focus on this image right now, this one. So they see a annular grating like this. Grating means nothing but black and white stripes, and they start. So this is the let's say this is time axis. This is a time. This time axis is same as this time. For now, you just remember, like, as time progresses, what is happening to stimulus contrast is A here. So here the stimulus onset happens where this kind of a grating appears. This appears, and then the contrast is slowly reduced. This is a time course of how the contrast is reduced and then increased. So here the participant's task is to respond whenever they see a change in they perceive a change in contrast of the given grating, and the beep shows their reaction times, how like what is their, how quickly they responded whenever they detect a change in the stimulus contrast. Okay, is this A and B part clear? So now we'll take this task, and while the participants are doing this task, they recorded their EEG signals. Okay, and we can this distribution can be divided into three parts. This is faster reaction times, medium reaction times, and slower reaction times. Okay, so their color coding is depicted by this legend: red, orange, and yellow. Okay, so now what they do is, on this participants, they compute an ERP from centroparietal electrodes. Let's say they take some central 3 electrodes on the scalp, and they take an average, and like what we do for ERPs, they plot an ERPs across trials. So you see that on fast reaction time trials, when they split the trials into faster trials in which participants responded fast, trials in which this is fast, this is medium, and this is slow. You can see this ERP peak, like this is a stimulus onset, and this ERP peak is informative of how their response is. Like if they're responding quickly, the ERP peaks in the mean of this vertical lines represent mean time of that fast reaction times. So all fast reaction times clubbed together, you get one mean reaction time, and so on for medium and slow. So, and also they can do an ERP image for better visualization. Instead of averaging across trials, we want to see whether it is happening on every trial or not. So they plot an ERP image here, has shown this graph you see. So what they're doing is, based on the reaction times, each trial is sorted. Like trial 1 might have faster reaction time, trial 2 might have very slow reaction times, but they sort all those trials such a way that the reaction times are sequentially arranged based on their how fast they. So if you take this trial number, this is a faster reaction time trial, and this trial is the slower reaction trial, and they plot an ERP image for that. And yes, so now what you see is this ERP, like you can see a dark patches here, right? Like this, the sigmoid shape thing here, like a hysteresis curve here. So this shows that this CPP, the centroparietal ERP, is informative of their reaction times or decision perceptual decision times. So this is one example to give, like how ERPs can be related to some kind of process going on. And when they average for each, you get a topo plot like this in this windows, like just this window they take and average and plot to see which uh, part of cortex is giving most response. Instead of taking just CPP, they're doing an ERP image, and here you see that most comes from this centroparietal region. From this is the occipitoparietal region, the second half, lower half, and in that you see the parietal region giving the highest response. So this shows that for this kind of perceptual decision process, this centroparietal ERP is informative, or it's a signature for perceptual decision making, because it is varying with one of the task parameter, that is the decision time. Is this clear?
Could you remind what does the black line represent? And 0, I assume, is the stimulus onset.
Yeah, 0 is a stimulus onset, and this line is if you make a cumulative distribution function of this reaction time distribution. Basically, just it is the actual time when the subject gave a response. So this black line corresponds to, on that particular trial, if you take, let's say, this trial, on this particular trial when that subject gave the response. So yeah, cool. So this is one example to show how ERPs can be used to inform task variables. Okay, I hope most of them are clear. Or if these plots are completely new, it's okay, you can ask if you don't understand any part of this. Okay, so basically, to summarize, we were looking at 3 different types of visualizations for ERPs. One is you can visualize for each electrode how the time course of ERPs change by averaging across trials for each electrode. And you can also do, in a given window, or if you want a component of interest on ERP, you can see how that component corresponds to cortical surface, like which part of the cortex is giving most response, by using topo plots like this way of visualization. And another one is to visualize trial wise time course and average in smaller number of trials, so that you get an ERP image. So yeah, so these are the 3 types of ERP visualizations. We will have practice in the next later sessions. Right now we just practiced only this part, the topo plot and this, but we'll look into ERPs in the later classes. So yeah, now just recap of visualizing frequency information. So like we discussed in the last class, this is the time signal. It's a 2 Hz sinusoid, and when you do a Fourier transform on this signal, it converts time domain to frequency domain. So this x-axis is frequency axis, and you can see at 2 Hz here, you see a peak. This is a frequency, this is time. So 2 Hz here represents how many cycles are going on in 1 second. Like if you take this 1 and 2, so this is first cycle and this is the second cycle in 1 second. This time is in seconds. So you see in 1 second there are 2 cycles, and when you do a Fourier transform, it gives a peak at that 2 Hz. So, and similarly for 2 Hz, for 3 Hz, and if you do a sum of signal and take a Fourier transform on this whole signal, you get 2 peaks. One is at 2 Hz and one is at 3 Hz. This is frequency axis. So this is a visualization of very simple signals, just sum of 2 sinusoids. Okay. This is one form of visualization, but if you have an even complex signal like EEG, it doesn't look this simple. You'll have a more continuous and complex spectrogram. So if you see a typical EEG spectrogram, it looks like this. So it doesn't look like you have nice single peaks, because this is not monotonic signal. This is a mixture of many frequencies, therefore you see thing like this. So this is from different electrodes. This is from P11 electrode, this is from P9 electrode, and this is from P6 electrode. So yeah, and yeah. So now there are another type of visualization where what you can do is you can cut the signal into segments. You can cut this signal into multiple segments and see the frequency content in each segment. So this gives a two-dimensional signal like this, as you see here. So right now you focus only on this part. This is called spectrogram. So here on x-axis is time and y-axis is the frequency in hertz. So what you see is in this particular segment of time, let's say this segment, this whole segment, so you can divide into time segments. In this segments, what is the frequency content? So whenever you see a frequency content here, very like I don't know if you can see the color, but yeah. So this part you see, right? There is a very high power of this frequency at that in time. So you can split into time and also visualize. So and when you average it, right, you get this kind of a spectrum. This is what we were seeing right now. So this is the frequency axis for this green plot here, and this is the power. So yeah, so this is what we are looking at. Is this clear? Is this kind of visualization clear? So this is nothing but just, you know, in segments of time signal we are seeing what is the frequency content. So these are the two visualizations of how to see a frequency content or spectrogram or spectral content in a signal. Yes, please.
Hi
Shubham.
Deepak, I
Yeah.
just wanted to ask, in the spectrogram, like, is the yellow color meaning like those frequencies have higher power,
Yes, yes,
and
yes.
and as it goes to black, it's like
Yeah.
lower power, right?
Correct. So there is some background noise, but this you can see. So I'll show a demo on like how we can interpret. So this might be a little fun, but let me see how to do this. So is this visible, the plot on my screen? Yeah. So second, is this not moving? I'll just reopen it 1 second. So there is this app which can analyze in real time. Okay, I think this is better. No, yeah. Okay, so I think this is not playing because I'm sharing my screen. Okay, we can come to this little later, but yeah. So what happens is if you talk or sing some monotonic tones like the notes in music like sa re ga ma pa, you see a nice steps going this way. Like you see, let's say this is time. Let's say you sang sa in some time and then re, it goes up and so on. So when you sing notes, no, this will be corresponding to sa, re, ga, and so on. Okay, I can show this demo a little later. There is some technical glitch. Some of that is not being played here. Okay. So now coming to a typical EEG spectrum again. So here you see a typical spectrum here, and we have divided the frequency axis into different bands. Delta band, theta band, alpha band, beta and gamma bands. And you see like different electrodes have different courses of or different frequency contents, and here you see this electrode P11, the green one shown here, has a very good alpha peak. So we say that this signal from this P11 has a alpha high content of alpha frequencies, and in the blue signal you see some peaks in beta band. So yeah, in low beta and high beta, low beta, high beta. So these are informative of some feedback processes from prefrontal to visual cortex. So yeah, but one thing you notice here is a common pattern across any EEG signal or any biological signal. When you take a spectrum, when you take their spectrum, no, so you see a decay like this. So initially at lower frequencies you have like a high power amplitude, and then it decays like this, right? uh, can you guys notice this decay? So this decay happens because of a noise called pink noise or fractal noise. Okay, so this happens because of how the biological systems are designed in general, and there are so many systems in nature which are designed this way, which causes this pink noise or fractal noise. We will look into its sources soon. So here, as I told you, there is this typical EEG spectrum with decay in, has the frequency increase. So this is also called 1 by f noise, and in EEG we say 1 by f, but ideal scenarios it is 1 by f power alpha. It can be any powers of f. So it can be 1 by f noise or 1 by f square noise, so on. So why this happens, we'll take a small toy example. Let's say this noise also happens because of a phenomenon called scale free network or scale free dynamics. So in general, if you take this kind of a network structure with nodes, you see there are very fewer like the large nodes are very few, but small nodes are many. So the number of small nodes are high, but the number of larger nodes are less. So similarly, here what it corresponds to is in your brain, right? Let's say your brain has a set of oscillators, and it has different bands of oscillators. There are oscillators are, let's say, some devices in your brain. They are there, and there are many oscillators which are oscillating at lower frequencies than the oscillators which are oscillating at higher frequencies. And because of this mismatch in number, as you go to higher frequencies, the number of oscillators decrease, and this causes this kind of noise called pink noise. So we can even simulate this noise by using this kind of scale free network or scale free dynamics. So it is also like, for example, if you have a class of students, and students who get higher marks are very few, but as you go to like lower end, of course it is more Gaussian, but to some extent the average marks, the number of people having average marks are more. So that is also like if you limit that range of marks, you can see even that has a scale free dynamics. So similar thing goes to the brain. Another real-time example from given by Mike X Cohen. You can look into his videos. They're very good. So here what they're doing is earthquake. On y-axis it's the earthquake magnitude, earthquake per year, earthquake frequencies, and on x-axis you see the magnitude on the Richter scale. That defines how big is earthquake. So here, as the magnitude increase, right, the higher magnitudes have lower frequency of occurrences. So similarly in spectrogram, the higher frequencies are less common than lower frequencies, and therefore you get this kind of a pink noise. Is this clear? So yeah, 1 second. Yeah, so yeah, is my screen gone? Just give me a minute. Yeah. So one second. Yeah. Okay. So here you see this kind of similar scale-free dynamics appearing in case of earthquakes. So is this clear why the, like, although not the deeper reason, but on the superficial level, like what could cause this pink noise? Oh, I'm not sure I fully understand. Okay. So imagine like if you, I'll explain through this earthquake. So as you go on higher, right, the number of earthquakes occurring in an year with a magnitude of 7, okay, yeah, with a magnitude of 7 is lower here. Yeah, if you take if you want to count number of earthquakes with magnitude 7, they're very less. This is on a log scale, but just remember it is very less in number. But if you take a smaller magnitude earthquake, they occur like almost so many times. Like there are always some kind of earth movements happening at this scale. So basically small things occur large number of times, large magnitude earthquakes occur smaller number of times. Similar to this, in the brain, the number of oscillators at lower frequencies, like number of oscillators at 10 hertz and at lower frequencies are more in number. So the contribution to that frequency will be more because there are more number of uh, like the energy will be more if you average those numbers. But if you take a higher magnitude things or higher frequency, the number of higher frequency oscillators are very few in the brain, and therefore you get a lower energy content in those higher frequencies. So sometimes this is a noise, sometimes it can be used like if you're using a longer scale, but usually it is considered as a noise because even though there is peak here, the magnitude has reduced because of 1/f noise. So what we do is we have to normalize based on some broadband power across all the frequencies. You take a mean and then you average so that everything comes to the same level. So and then we tend to compare, like, which, if you want to compare two kinds of oscillations, we first have to normalize the spectrum and then compare. So otherwise you're comparing apples to oranges or something of that sort. So okay, cool. Yeah. So if you see, this is how a general EEG or any biological signal looks. But if you take white noise, it has a uniform frequency content. You don't, like, if you simulate a white noise, it will have a, like, it will not have a decayed kind of thing. But typical signals look like this, and they have a, at lower frequencies they have high energies, but at higher frequencies they have low energies. Yes.
There are some questions.
Yeah, yeah. Can you please read once?
So, one question is that what are oscillators in the brain? Are they group of neurons firing at particular rate?
Okay. So this, I mean, the oscillations that you see is because of the oscillations going on in a population of neurons. So sometimes a set of population can synchronize their, it is not spike rate per se. It is, there are electrical events which are not spikes also. So they have small voltage changes, like post-synaptic potentials and all. And if a population has, if a set of population of neurons has similar kind of electrical activity below that spike rate, uh, below spike threshold, those create these kind of oscillations. So it's basically interaction of neurons creating these oscillations. Yeah. The next question.
There is another question that what do higher degree oscillators reveal, and where can they be found in the data set?
Oh, you don't have a like I just gave an analogy of oscillators in the brain, but like every kind of oscillator is there in the brain. So it's like which frequency content is higher or lower, and you generally people analyze this gamma frequencies, which are higher frequency bands. uh, can I see the question? 1 second, just give me a minute. Yeah. So what do the higher degree oscillators reveal? Data set, you just have to plot the spectrum and see how the spectral content is. But when you try to like, if your interest is in gamma frequencies, it is always good to normalize the, basically to remove this 1 by f noise, and only then you can see clear picture of what is happening in higher frequencies. Okay, so yeah, so Shubham would ask another similar question but more specific here. He's asking, what do the what do special properties do they give to information processing in neural networks in the brain? Okay. Why are many biological networks scale free? It is scale free. It is just how the design is. So I have no specific answer, like exact answer why it is, but the structure of brain is arranged like that. But this high frequency, this later frequencies actually are used in longer time scales. So for example, if you're doing some aging research, they see that there in those cases 1 by f noise becomes more important, because uh, there is a paper I can share later, but in that research what they found is the 1 by f noise is actually different from younger versus older populations, showing that the slope of the rate of 1 by f fall is slower in the case of aged populations, but younger populations, they have a uh, faster faster 1 by f fall. The 1 by f fall is faster. So if you're analyzing in that case, this becomes 1 by f noise becomes a feature, because uh, it's saying something about the structure of the brain. So the, like I told, the number of oscillators, so the structure of the brain is changing, therefore the 1 by f fall has changed if you take on a longer time scale. So is it clear, Shubham? Yeah. Good. Okay, so this also partly answers what do high degree oscillators reveal. So they're also like they also analyze gamma frequencies in case of gamma bands, but it's very rare in EEG datasets because of higher 1 by f noise in the EEG. But if you take a LFP signal, right, the population of neurons that you're analyzing is smaller. There the 1 by f fall will be less, and the LFPs have uh, the 1 by f noise is smaller in that local field potentials, which are analyzed using depth electrodes by inserting electrodes in the cortex. So I hope it's clear now. Yes, sure. So coming to the last topic, I think I took 40 minutes, but it's fine. Yeah. So as I told you in the last class, EEG has a very poor spatial resolution, and a dipole at some point can cause a voltage change at any point on the scale, although it is like the amplitude of amplitudes will be higher for that potential when it is closer, because it is inversely proportional to the distance from the dipole. So closer distance have uh, higher activity, showing more effects of that particular dipole. But let's say there is a dipole here, this the one which I marked green right now, and this can cause a potential change at this electrode, and at even the farthest electrode. Similar thing happens for a dipole taken here. So localizing what exact source in the brain causing this kind of a potential is very difficult with EEG. Therefore, we usually complement with fMRI studies, or other stimulation studies where you silence some region and then see how the potentials are changing, or you see neuroimaging of two kinds simultaneously. So it's called simultaneous EEG and fMRI, where you see internal activity going on and the scalp potentials going on. So there you have a multimodal data sets where you can compensate for disadvantages, advantages, or limitations of each other. So with fMRI you get a good spatial resolution with EEG, but also there are few techniques which are useful to localize the source of EEG. This is called source localization problem. Although it's an ill-posed problem, we will what is meant by ill-posed we'll take it later, but yeah. So there is no exact solution. That is what meant by ill-posed problem. So you have some techniques which are approximations, or using some assumptions you approximate the sources. So that techniques are called source localization techniques. So instead of taking channels, you find the sources of particular voltage time course or potential time course, EEG potential time course. So why does volume conduction happen? So first thing is we are measuring very far away from the source, and also the the layers of the skull, or the meninges, then the skull, all this are not isotropic. They have different different characteristics at different positions, and therefore an electromagnetic signal propagating have different attenuation levels at different points, and also they're like a conducting medium, therefore they pass through whole the brain, and you see this volume conduction effect happening. So there are few techniques which uses finite element method, like they draw small mesh grids over the brain, and in those each mesh grid they use some matrix theory based analysis to find the sources. And so why this is a big limitation for EEG? Because for example, let's say you're measuring how two regions are in synchrony or communicating with each other. So like whenever two regions communicate, they are mostly in synchrony, the activity of those regions are in synchrony. And let's say there are two dipoles. Because of this dipole here, you get a good signal here, but a weaker signal on this part of the scalp. And similarly, you have another region on the right side of the brain, and similar pattern you get here, one signal, and on this electrode one signal. Now in EEG what you're doing is you are comparing these two electrodes, how they are synchronous between electrodes. So you'll get synchrony automatically here, because the same activity is going here and here. So this will be summing up and this will be summing up, and you see like perfect coherence. But the problem is their actual coherence is one problem. So when you're analyzing this kind of metrics, you have to be very careful and minimize the effects. So what you do is, there are techniques to minimize this. It's called current source density. So what they do is they take difference between neighboring electrodes and subtract that signal. For like let's say one electrode you choose and there are 4 neighboring electrodes. You take the average of this and subtract it from this electrode. So that will make the sources more orthogonal. So we will discuss that in more detail, but Yeah, there are few techniques. This is called Laplacian operator. So on the EEG signals, you use a Laplacian operator to reduce the volume conduction effect, and then you try to analyze this kind of metrics. Or whenever you're analyzing between two regions using EEG, you have to be very careful, or saying that this signal corresponds to this region. You have to take it with a grain of salt. So this just shows one of the main limitation of EEG. Yeah. So yeah, so there are multiple techniques on finding sources. So you have this EEG responses. So from some brain activity is causing this kind of activity, but from this activity, how do you go back to sources? This inverse problem is called inverse problem. When you solve this problem, we call this as source localization. That is, from EEG time series, you are trying to find what are the sources of this, and there are many techniques, not one technique, and each technique gives a different kind of uh, result, because there are some inherent assumptions within them. So this is just to depict, don't have to worry about the details, that yeah. So not just electrode space, we can also do source localization and find how each source is causing certain potentials. So yeah, is this fine? Any doubts here, or source localization, volume conduction? Okay. So yeah, I think I can take this in the next session when I come back to signal processing. So right now we'll, if you have any questions, you can text, or we can start with Atharva's fiber photometry introductory lecture. So yeah, over to Atharva. If you have any doubts, please message, or you can unmute and talk also. This vocal is still not working. Yeah, I'll come back this to the end of the lecture if time permits. Yeah. Yeah.
1 minute.
Yeah.
Mm, I'm just trying to present it on my screen. Okay. Okay. Are you able to see the screen now?
Yes, Atharva, we can see. Yes.
Okay. So, hi everyone. I am uh, Atharva Modi. I'll be presenting introduction to fiber photometry in today's lecture. Somehow I am not able to see my mouse myself. So I was thinking of uh, going to the pen. Okay. Yeah. So before we start, we covered uh, EEG in previous 2.5 lectures, which records brain activity using by measuring electrical activity on the scalp of the person. Uh, so I want some answers from you before we proceed to another technique. Like what all techniques uh, can you think of when you want to measure brain activity? Some of them are already on the background of my slide. I want the answer light as the last of them. So when you think that you have exhausted all the other methods, say light. So I want answers from all of you before I proceed. Like, what are the ways in which we can? What are different ways in which we can measure brain activity? One of them is measuring electrical signals.
We can try MRI. Yeah.
Yeah. Other is using magnetic uh, fields in MRI, so that is a very indirect way of measuring brain activity that measures the level of oxygen in brain,
Himanshu
in the
says
blood
FNRI.
vessels around the cells. Others are TDI. What is TDI? fMRI, FNRI. Oh, okay, yeah. Near infrared spectroscopy, if I am not wrong with NIRS.
Yes.
So that again is not a direct measure of like neural activity. Uh, and with NIRS, like it's near infrared spectroscopy. So you send some near infrared signals in the brain and then measure the output. Uh, so it's a non-invasive technique again, which can give you information about different metabolic states of the brain tissue. EG, ECOG, again measuring electrical signal. So we are measuring electrical signal. We are measuring magnetic signal, magnetic state basically. We are measuring uh, in spectroscopy. We are actually sending electromagnetic wave, light, which is not light but near infrared electromagnetic wave, and getting some emission out of the brain and then studying that. Track density imaging. Okay, that track density imaging, I'm not fully aware of what that means, but if I have to guess, it's something related to the tracking of fibers in the brain. Uh, Calcium imaging, yes, calcium imaging. You are using now light to measure the activity inside the brain in calcium imaging. So there are of course other way, like just there are invasive way in which you can just put electrodes into the brain. Again, electric uh, uh, electrical measurements are there. There are other measurements using light instead of imaging. So when you send light signals, electromagnetic wave signal inside the brain, it produces heat inside the tissue. So these are very recent developments in the field where this heat is then converted. This heat also results in vibrations inside the tissue, and which ultimately results in ultrasound signals which we can capture from outside the brain. So these are all various different techniques. But what I am going to cover is also very innovative, a very intriguing technique where we can record different kind of brain activity. Uh, we'll see what different kinds mean using light. So by shining light inside the brain. So uh, uh, yep. So we saw this image now multiple times when Deepak was presenting and in the introductory lecture. Uh, these are all different spatial and temporal scale and different kind of techniques that are available to us to study functions of different kinds of function in the brain. What is fiber photometry? Photometry falls somewhere here. So it is an invasive technique to measure brain activity using light. Invasive means we have to go inside the brain, and the spatial and temporal scale fall somewhere here, around 500 micron spatial resolution, which include hundreds to thousands of neurons. So it's a bulk activity measurement, and the temporal resolution is around 10 milliseconds. So that's like uh, a few action potentials. So it gives us quite good temporal resolution, and uh, not bad temporal resolution, not very good temporal resolution, but the spatial resolution also can be improved, which will I will mention how do we do that to individual cell levels. This technique in general provides a very lightweight on the animal's head, and it's a very not a very very light hardware and data light data technique to measure brain activity. So, what do we need to record from the brain directly using light? Uh, of course, we need a biosensor, right, which responds to light, and we get some output out of it, uh, which scales with our analyte. Analyte here is the thing that we want to measure, right? It's called analyte. So we can use fluorescent proteins. So these are the wonder of 1980s, I guess, which were discovered in biological systems, especially in seawater, jellyfishes probably. So these fluorescent proteins have these properties to whom you, like people might not know this, that there are these proteins on which if you shine some light, uh, they emit a different wavelength of light, basically by Stokes shift. It shifts to a larger wavelength, and it emits that wavelength. So this property of molecules is called fluorescence. So if you want to utilize these fluorescent proteins to measure brain activity, what properties do you need in your proteins so that it enables you to record brain activity? Any answers? Like what kind of properties can you modify in these kinds of protein? Basically, what do you want? If you want to measure brain activity, what do you want from these proteins? So that you get some relevant signals out of them. So, any guesses on that? Can speak if you want, or you can type.
Can I?
Yeah, sure.
Okay, someone says Shubham Hotasis binds to molecules involved in generation of action potentials.
Yes, exactly.
Yeah.
It should bind. Uh, that's one thing necessary. But cell biology interacting with these proteins are like similar answers, but this interaction should result in some change in the output here, so that this measure somehow depends on the thing that we want to measure. So let's say if you want to measure voltage inside the brain region, then this output intensity or whatever properties of this output should change based on that particular measurement you want to do. So if let's say there is high voltage, this intensity gets high, or if there is low voltage, this intensity gets low, something like that, right? The wavelength should change proportional to brain activity. That is one way. The wavelength can change the fluorescent wavelength, but the most generally occurring, so what people have made is genetically encoded fluorescent indicators. So these are like similar kind of like you take GFP and modify it. This is a sequence of peptides like it proteins. So you modified it in a way that it now interacts with uh, the analyte. basically the thing that you want to measure. So let's say if you want to measure uh, concentration of some ligand or let's say neurotransmitter, dopamine, inside the brain. So what you want from this genetically encoded fluorescent indicator is that when in presence of analyte, the analyte can bind to this particular indicator, and now this indicator fluoresces much more than what it does without the analyte, right? So what Himanshu suggested was that the wavelength should change, but what people have what people have achieved in the field of molecular biology is to create this genetically encoded fluorescent indicators, which doesn't change wavelength, but which changes the intensity of emissions based on the analyte's presence or absence. So what we are doing is shining the same light, reading the output in the other emission wavelength, and this emission wavelength now can serve as a proxy for the concentration of analyte, right? In the presence of analyte, the emission intensity will be much more than in absence of it. So one of the examples of this ligand-dependent fluorescence sensor looks like this. So this is a membrane and this is a G protein coupled receptor. You don't need to know what it is to understand this. So this is a receptor basically which binds to some ligand. Now this ligand can be any neurotransmitter in this case, like dopamine, like glutamate, like serotonin or norepinephrine, anything. This ligand depends on the properties of this receptor. Now this receptor has this modified GFP, this circularly permuted EGFP. That is again, this is a genetically encoded fluorescent indicator, GFE. So this is a property that when ligand binds to it and you shine light on it, this emits wavelength, a green color. So you can measure this green color and it indicates the presence of ligand. So this is the special property that these kinds of fluorescent indicators have, and that enables them to serve as a biosensor, right? So the fluorescence increases as the concentration of ligand increases. Now this concentration of on the x-axis, you can design different kinds of proteins. You can design proteins which vary fluorescence based on concentration of calcium ions, which is not a ligand, it is ion. You can change on the basis of voltage, or you can change on the basis of different chemicals presence. So now we know that when action potential occur inside a cell, the calcium ions uh, increases very rapidly inside the cell, from extracellular and from intracellular membranes also, like vesicles also, they release calcium ions inside the cytoplasm. Now if this kind of indicator is present in your cytoplasm and an influx of calcium happens inside the cytoplasm, then you will get an increased fluorescence overall. So if some cells of that area fire, then you will get an increased fluorescence. If all cells fire, then you get a very high fluorescence. So that is the kind of characteristics these biosensors have.
Atharva,
Yes.
can you show this a neuron with this membrane once? I don't know if people are aware of how a neuron looks or what this membrane is, if possible.
Means
I hope they know, but I'm just thinking if they're not.
means these kinds of, this is a fluorescent indicator biosensor, right?
Okay, it can
So
be
you
any.
need not express it on the membrane first of all.
Okay.
It can also express in the axons or synapses or anywhere in the cell basically.
Okay.
You get so if you want to measure, let's say, concentration of dopamine at the synapse, So you want this protein to express at the synapse and not in the uh, cell body, basically. If
Okay.
you have a neuron, so neuron looks like this is the cell body and this is the axon. So if you want to measure something here, then this receptor should express here,
Okay. Okay.
this biosensor,
Okay. Got it.
not in the cell body. So the important thing is that we should have this kind of fluorescent indicator inside the uh, brain and in the regions where we expect the analytes, whatever we want to measure, to change,
Okay.
right?
So, can you just explain what is this blue waveform here? This is
This
a light from external sources, or?
this is what we are shining. So you have
Okay, okay, okay.
basically any source of light which you shine on the
Oh, on this protein and this
cell,
diff. Okay, whenever
and
the.
it will emit a different wavelength.
Okay, okay. Fine.
So that's the property of this fluorescent proteins.
Okay.
But if you shine the same light here, you will not get that emission.
Okay.
So no emission.
Okay, okay. So that ligand binding is very important to emit some.
Yes, yes. That makes it a sensor for this particular ligand,
Okay, okay.
right?
Yeah, yeah.
Now, as Deepak also pointed out that this light we are shining. So the fluorescence is also a function of light, right? So the more light we shine, the more fluorescence we get. So in general, we don't consider, this is just a note on this, that we don't consider fluorescence as the signal, we consider the change in fluorescence as signal. Uh, and for example, so this is a one type of biosensor for dopamine, D light. So this is the plot for fluorescent signal with concentration of dopamine, right? So here the plot is delta F by F, basically the proportional change in fluorescence over in the fluorescence with concentration of dopamine. So you see that there are different variants of D light, and each of them behave differently in presence of dopamine. So it is you can basically measure this quantity using light, and you can get an idea about the concentration of dopamine present in that particular region of the where you want to measure the concentration. So to choose a different kind of sensor is another thing that we need to consider here. So you need to look graphs like this. You need to look at the value of KD, which is the dissociation constant. So this dissociation constant uh, means that at what concentration of dopamine you achieve 50% of max signal. So 50% of max. So basically, this is the max signal that it gives. So around 9. So this is 4.5. This is the KD point for this D light 1.3B. So these are different variants of protein which show different KD, and higher the KD, lower the affinity for ligand. So if you want to measure somewhere in this range, in the lower range, you need a very low KD. So with delight
Hello.
1.3b, you can't measure these ranges of concentration. But with
Hello,
delight
Ashwin.
1.4, you can measure these ranges.
Yeah.
But you can't measure these ranges. Basically.
You want to
So
come to Mac?
this guides the choice of the sensor that you want to use.
It's
This
Atharva
this
teaching.
this
I'm
was
just a
about
moderator.
the variation with concentration of neurotransmitter.
Hello.
But now, what about timing? So this temporal scale,
Okay.
what does it depend on? Like, can anybody from you point out, like, this is the signal, delta F by F, and this is 500 milliseconds. It's so fast. Like this happens within a few less than 10 milliseconds it rises and then it shows a falling characteristics. So if I want to say that what kind of temporal resolution this technique have, what do you need to look into? So, and beware that this rising characteristics may be different from falling characteristic. Even if you have a step change in basically dopamine, these these characteristics might differ in rising and falling. So, will anybody of you like to answer like what does it depend on these characteristics, and what do we need to consider about the sensor that uh,
Mm,
okay. So basically, it depends on the kinetics of the the indicator and ligand interaction. So basically, if kinetics are fast, it shows a very high rise time and very fast low, fall time also. So what it indicates that DA binds to the ligand, DA binds to the indicator very fast, but it leaves the indicator slowly. So that's what generates this kind of uh, profile for this del F4F. So this is something we need to consider while choosing the sensor. This is overall gist, but the thing is that these kinds of proteins are not expressed inside the brain. So how do we get them into the brain? That's one question. So we can measure different kinds of neurotransmitter or activity level inside the brain using these, but how do we get them inside the brain? Uh, Anybody can will like to answer that? How do we get these inside the brain? It's very simple. Yeah, through surgery, but like do you inject the protein or uh, so many people are not from biology background. So basically,
virus.
if you inject protein, like it will get metabolized or it will get degraded inside the cell over a few hours or days. But what you want is expression of protein in that, but in your region of interest for longer time. So what you do is you inject the genetic code of the proteins inside the brain. So what you do is you pack this genetic code in some harmless viral vector and then deliver it to the region of interest using an injection. So this genetic code, once inserted, gets expressed. You can design it in such a way that it expresses uh, for long times. Like it expresses continuously in the cells that the virus infects, and that's how you get these proteins to be expressed in the in region of interest inside the brain. So now you have the biosensors in place. So once you have the sensors, what do you need next? So you need a measurement device for the sensor. So now they are effectively genetically modified rats. Uh, uh, yes, but not all cells of them are genetically modified. Like we are not changing the DNA, but we have expressed some kind of um protein in them which is not naturally present in particular part of the brain tissue. So these are not these have genetically, like these have unnatural elements inside their brain. That's it.
So is it like just the extra characteristics coming, like extra that protein coming in, but not changing the DNA or something?
No. So what we do is uh, this genetic code is changing the inherent DNA of the present in the nucleus, but they go and
Okay.
this is also DNA only,
Okay.
but a piece of DNA that is expressed on expressed separately.
Okay.
Basically, it is not part of the uh, main uh, double helix DNA that the rats have in their nucleus.
Okay.
So they sit in the cell. So the virus has the property of infecting the cell. Basically, it enters the cell, and this uh, genetic code gets into the cell, and now it starts expressing. Once it starts expressing, the protein starts producing, and it produces. It basically replenishes itself. That's how it works. Now it is. So once we have sensor in place, we need to deliver and record light inside the brain. Right? So how do we do that? We implant a fiber uh, inside the brain using the same stereotaxic surgery. We take an optical fiber and glue it to the skull surface. So this prevents the movement of the fiber with respect to the skull, and it stays in the same place as we want it to be. Let's say we want to measure from hippocampus, we'll place it above the hippocampus to certain depth, and then shine light here using a optical fiber. So that's how a rat looks like uh, when we actually do the experiment. So now we have sensor, we have the light delivered to the sensor, we have light coming out of the sensor. We need a uh, measurement device, like how to quantify this, but how to measure this emission intensity. So what it looks like is that there is a light source. So this light source can be of multiple wavelength, or you can even have a green light source basically, or whatever wavelength you want it to be. So this is a light source which gives out a particular wavelength of light which is shined on the brain, and some emission light comes out, which is again passed through an emission filter. You want to measure some particular wavelength. Basically, in our case, it was we were shining blue light and measuring green light. So here they are shining green light and measuring orange one. So now you can measure this using multiple uh, like there are many options available here to detect. One of them which is very reliable is photomultiplier tube. Another is like photodiode, or you can install a camera here which detects the light intensity coming out of uh, this particular fiber. So now you have everything in place. You have basically light source, some optics going through the animal. The emission light goes to a light detector, and now it digitizes the signal, and we get some signal like this over time, and then we can pre-process and analyze these signals. Uh, so what you can do is you can have this rat in a behavioral box. Basically, you can do any kind of test on it, and you can integrate this behavioral electronics with the photometry electronics and do the time synchronization here. So for those who are not who are not familiar with this, that so this particular detector and digitization and all the electronics here work on some clock, and that clock is independent of what clock works is measuring the behavioral events, right? Both are on different clocks. So what you need to do at every measurement is that you need to send time of behavioral events in this. You need to synchronize both the clocks basically. You need to know when here is when it's 7 millisecond here. How many milliseconds have passed in this clock? So you can calibrate both the clocks together and then get signals for each behavioral event. And yeah, this is very similar to event triggering in EEG. Very like it, you just you can trigger it in various ways. Yes, similar to event triggering in EEG. Yes. Now there is this one important consideration that we will consider before going on to the signal processing part of it. So the consideration is that this particular optical fiber that you have installed and you are measuring some light out of it, how are you sure that any changes in that emission light are due to changes in the concentration of uh, dopamine or concentration of calcium signals that you want to measure? So, so we are not sure. We need some test for that. We need some control for that. This change in emission light can also happen because of movements. Yeah, these optical fibers are moving components. Uh, so that particular movement can cause some kind of changes in the emission intensity. There are artifacts here also. Brain tissue itself have some movement or some changes in the blood flow or something, which can cause the change in intensity of emission light. Are rats the typical model system for this technique, or could you use smaller organisms? So uh, we can use rats, mice. People do this kind of technique in uh, like in vitro cells also. You can do it in slices, brain slices also. What you just need is a genetically encoded sensor which expresses in the cells, and you keep the cell live. And if you, let's say, electrically stimulate it, the upstream or downstream of it, and then you see what kind of changes the calcium levels or neurotransmitter level happen in that particular cell. So you can do it in any model system basically. This is done even in plants. So people have measured the calcium waves in plants. Probably they have used some other kind of sensors, but you may find very interesting videos of plants communicating each other with calcium signals. It is very similar to this fluorescence imaging. So yeah, what I was talking about is that this emission light's intensity depends on not only on like can change not only on the basis of uh, the concentration of the thing that you want to measure inside the brain, but also due to some external factors. So what kind of control do you have for this? It's very interesting thing. So we saw this that this particular indicator works like shines brighter in presence of the analyte you want to measure. So this also have a different like a separate property. What it does is that if you shine a different wavelength, so here I was shining, let's say, blue, and now I'm shining UV light. So if I shine the UV light, it emits the same wavelength, but this particular wavelength this particular emission intensity when I shine the UV light, it doesn't depend on the analyte. So this is a very intrinsic property of the designed protein. Most uh, most encoders will have this kind of property, that there is some excitation wavelength here. So this is blue one is called excitation wavelength, and this UV one is called isosbestic wavelength. I'm calling them blue and UV. They may not be necessarily blue and UV for all the different kinds of sensors. And this green one is called emission wavelength. So most biosensors have this property, that if you shine isosbestic wavelength, it does not depend on the analyte. So basically, any change in the emission wavelength when you are shining the isosbestic one, uh, that change is not due to the presence of analyte, but due to the external factors. And we assume that these changes are proportional. So any change due to movement will be proportional in both the cases. So that's what that assumptions makes it possible to control for other artifacts which affect the output. This is an interesting way to deal with the external signals affecting our emission intensity. So now I'll go through some like what we have seen, like advantages of fiber photometry. Like we can measure deep tissue bulk activity unlike EEG or other non-invasive techniques. Uh, s- but it requires, of course, genetically encoded sensors to be put in the brain. Also, we can measure individual cells using add-on imaging techniques. So if you put a camera on top instead of a photomultiplier tube, so camera can measure now pixel wise, and the cells may be at you can use basically advanced optics to resolve the signal uh, in pixels, and you get cell wise signal. Basically, you can find cells out of it. And you not only measure the electrical voltage or ion concentration, you can measure all kinds of different things like calcium concentration, voltage. You can design proteins which are specific to some neurotransmitter, dopamine, oxytocin, norepinephrine, or serotonin, or glutamate. So these chemical concentration, being able to measure it at such temporal scales without having a bulk equipment like voltammetry or putting a chemical electrode inside the brain, this is very like this is a very big advantage provided by fiber photometry. Now if you compare this with other detection methods like electrophysiology or voltammetry, you will get that this is a cheaper method also, which can provide a decent temporal resolution with even it can provide till multiple cells, and you can even resolve single cell if you uh, apply a different like sophisticated optics to this process. And you can also get cell specificity. Basically, if you want to measure from let's say only neurons which have glutamate, so you can express these proteins only in the neurons which uses glutamate as neurotransmitter. Other neurons will not express this protein. So you can design, you can play around very much with this molecular biology techniques, and it can give you cell specificity, molecular specificity, which not many other techniques can give you. So this is the very big advantage of fiber photometry. Ligand binding to GPCR is essential for emission. What exactly triggered that binding that results in light emission even during muscle movement, etc. So the thing is that it may not be a signal from the biosensor itself. It may be the movement may create artifacts in your optical fiber, or in your that creates a false measurement. Basically, it creates noise in optical fiber that increases or decreases the measurement you have. So it's not the change in the sensor that is happening that causes the uh, increase. That's that's one answer to it. Now, yeah. So uh, this is about advantages. Now, in the next class, we will see like how to analyze with data. So from this class, I will just show that how does the data look, and from next class, we will study like how do we analyze this data. So this data looks like this. So we have excitation signal here, E. We have isostatic signal here, and we have this behavioral events in form of messages. There are different messages. So the second column is time in microseconds when you started the measurement basically, and this F value is the fluorescence that you measure. Uh, uh, for different excitation or isoasbestic wavelengths. So if you see, here we are using a strategy where they're alternately we are doing like at 0 time point we are doing excitation, at 5 milliseconds we are doing isoasbestic, at 14th millisecond we are excitation, at again after 6 milliseconds we are doing isoasbestic. So these are um, alternating between each other. So once we shine excitation wavelength, measure, then we after 5 milliseconds we shine isoasbestic, measure, then after some time we again shine excitation, then isoasbestic, then excitation, then isoasbestic, then excitation. So this is one strategy. People use different kinds of strategy to measure. You get F readings for both, and now you can see like how are these F readings changing for both of them, and get some inferences out of that. Another data point is the behavioral event. So these are different behavioral event you can code. Like 3 means probably rat did something, like rat licked the port, or 2 means that rat pressed a lever, and you get times in microseconds. So basically now you can sync. This is a sync signal. So you can get the activity around this point. Every point of this you can get excitation activity, isoasbestic activity, and compare both and check if there is something in excitation. So this is how the data look. Yeah. So I will show you a video of it before we leave for today. How does it look on the animal? I hope you can see my screen. So, and also hear the sound. I'm not sure.
Yes, Atharva, we can see.
Okay. So, so basically, this is an optical fiber on top of the head of the like inside the brain of the animal, and then it's doing some task, and we are continuously measuring the activity. You can see that bluish or UV tint on the optical fiber at the brain. So this is a pretty lightweight thing on top of the head, and it doesn't affect the rat's performance much. We are doing it under red light so that it doesn't affect the output. It doesn't affect the. So yeah, this is one kind of change that can happen. So if there is some light frequency, basically 50 hertz white light that is being leaking through the optical fiber, then you will get that signal in both isoasbestic and excitation. So that is one kind of error that we can control for using isoasbestic signal. So yeah, this is so in the next class we'll start analyzing the data for whatever I showed, like